Hello everyone, welcome to my channel Microbiology with Shabu. In this video we are discussing about the history of microbiology. So let's start. Discovery of microorganisms. Antony van Leeuwenhoek was one of the first people to observe microorganisms using a microscope of his own design. He called them as animalcules and made one of the most important contributions to microbiology. Spontaneous generation versus non-spontaneous generation. Theory of spontaneous generation or abiogenesis states that living organisms can arise from non-living matter. It was proposed by Aristotle and was widely accepted for almost 2000 years until flies infested meat experiment by Francisco Redi challenged it. Francisco Redi carried out an experiment on decaying meat and its ability to produce maggots spontaneously. Redi placed meat in 3 containers. One was uncovered, a second was covered with paper, and a third was covered with fine gauze that will exclude flies. Flies laid their eggs on the uncovered meat and maggots developed. Also, the flies were attracted to the gauze-covered meat and laid their eggs on the gauze. These eggs produced maggots, thus the generation of maggots by decaying meat resulted from the presence of flying eggs, and meat did not spontaneously generate maggots as previously believed. In 1748, John Needham carried out an experiment on spontaneous generation. He boiled the mutton broth, allowed it to cool and sealed the flask. Broth became cloudy and contained microorganisms, and he was the supporter of spontaneous generation. Lazzaro spallanzani improved on Needham's experiment. He first sealed the flask and then boiled the broth. No growth of microorganisms were found in the broth. He also found that boiling broth would sterilize it and will kill all microorganisms in it. He also found that new microorganisms could settle only in a broth if the broth was exposed to air. Finally, the controversy was resolved when Louis Pasteur devised a suitable experiment to show that it was the dust in air which carried germs that caused spoilage. He placed a nutrient solution in flasks, heated their necks in a flame, and drew them out into a variety of curves while keeping the ends of the necks open to the atmosphere. Pasteur then boiled the solution for a few minutes and allowed them to cool. No growth took place even though the neck of the flask were exposed to the air. Pasteur pointed out that no growth occurred because dust and germs have been trapped on the walls of the covered neck. If the neck were broken, growth commenced immediately. Germ theory of disease. Germ theory states that infectious diseases are caused by microbes. The role of microorganisms causing human diseases was first observed by Girolamo Fracastoro. He suggested that diseases was caused by the passage of germs from one thing to another, and he described 3 processes for their transmission: direct contact, indirect transmission, and transmission by air. He further recognized that disease-causing germs exhibit specificity, indicating that different diseases occur in different hosts and that different processes of transmission occur for different germs. Support to the germ theory of disease. Agostino Bassi showed that a fungus was associated with a silkworm disease. In 1845, Berkeley showed that potato blight, which caused the great potato famine in Ireland, was caused by a fungus. Louis Pasteur investigated the pebrine disease of silkworm and showed that the disease was due to a protozoan Nosema bombiasis. Robert Koch and his major contributions. The first direct evidence for the role of bacteria in disease was provided by Robert Koch, who showed that anthrax disease of sheep and humans was caused by the bacterium Bacillus anthracis. He had investigated the disease, isolated the pathogen, and confirmed bacterium as the causative agent. Koch identified the causative agent of cholera, the cholera bacillus, and named it VIBRIO cholera in 1883. He studied the life cycle of Bacillus anthracis and its causative role in the disease. He introduced the staining techniques and he demonstrated the method of obtaining bacteria in pure culture using sold media. Robert Koch's postulates. In 1884, Robert Koch proposed the following criteria, now known as Koch's postulates, to prove the causal relationship between the microorganisms and the host. The microorganisms must be present in every case of the disease but absent from the healthy individuals. The suspected microorganism must be isolated and can be grown in pure culture. The same disease must result when the organism in culture is introduced into a healthy individual. The same microorganism must be re-isolated from the diseased individual. Limitations of Koch's postulates. The disease caused by opportunistic pathogens. For example, many urinary tract infections are caused by Escherichia coli, a bacterium that occurs in intestine of all healthy individuals, but causes disease if it accidentally enters the urinary tract. The disease process involves cooperation between multiple organisms. For example, the disease impetigo, a skin disease, often is caused by the combined infection of Streptococcus and Staphylococcus. The causative agent cannot be grown in pure culture in vitro. Example, Mycobacterium leprae. The causative agent has no animal models. Example, HIV and Neisseria gonorrhea cannot produce their corresponding disease in animal. Koch's molecular postulates. Genes responsible for virulence should be present only in pathogenic species or pathogenic strains of species. Removal of virulent genes should reduce virulence of microorganisms. Re-isolation of virulent genes should reintroduce virulence of microorganisms. Thank you so much for watching my video. Stay subscribed for the next part.